Hey everybody, it's Dr. Gilchrist, and to finish up this week, we are going to talk about neurotransmission. Now, out of all of the topics that we are going to cover this week, this is probably going to be the trickiest section and the trickiest chapter. It's also going to be the most intense. So because of that, I really want to encourage you as much as possible to take a look through chapter 3 in your textbook. And remember that because these are recordings, I would encourage you to go back and listen to these as often as you need, because this will make up about 1/3 of your quiz that is going to be due on Sunday. So hopefully this will give you a pretty good overview. If you have any other questions, feel free to let me know at our weekly session, or shoot me an email, or put a post on the forums. So what are we going to talk about with this section, talking about neurotransmission? Well, we're going to mostly be covering 3 different things. We're going to start by talking about the basics of electrical transmission within a neuron. After that, we're going to talk about an overview of the neurotransmitter life cycle. And then I'm going to finish up by talking about some of the most common neurotransmitters that you are likely to encounter in a course based on psychopharmacology. So let's go ahead and start by talking about electrical events and electrical transmission within neurons. So generally, neurotransmitters, which are going to be really important for understanding how drugs work, are going to be released as a part of electrical transmission. So electrical transmission is basically a series of electrical events that occurs within a single neuron. It's going to start at the beginning of the axon, where the axon meets the soma. The term that we use for this is called the axon hillock, and then that electrical transmission and these electrical events are going to basically move down the length of the axon. So how do neurons ultimately communicate? Well, it's going to start with what is called an action potential within a single neuron. And an action potential is basically when the membrane's electrical potential, so a neuron's electrical potential, actually has a very rapid change. In this particular instance, the voltage begins as negative. However, through the events of the action potential, this voltage will shift from negative to positive within a millisecond or two. But to understand how action potentials happen, we need to first understand what is going on with a neuron at rest. So I want you to remember this number. -70 millivolts. This is the resting membrane potential. And basically, the reason that we have this resting membrane potential is because we do have a difference in electrical charge inside and outside of the cell. And because we have differences in electrical charge, we can basically say that the neuron's cell membrane is what we call polarized. So basically, at rest, the interior of the neuron is going to be negatively charged relative to the exterior. So basically, the inside of the neuron has more negative charge than the outside of the neuron. And this membrane potential at rest is set at -70 millivolts. And this basically results from the interplay of a few different types of components. So first of all, we have more negative ions, such as chlorine, and negatively charged amino acids inside of the cell. We have more positive ions, like sodium, outside of the cell. And additionally, what I will mention here is that at rest, the neuron is selectively permeable to different types of ions. So sodium, which, as we're going to talk about, is going to be a really critical player in this process, there's more sodium outside of the cell than inside of the cell. Because of that concentration gradient, sodium wants to rush inside. Additionally, Sodium carries a positive charge, as you can see right here, and it's very strongly attracted to the negatively charged neuron. But one of the things that we find is that at rest, sodium is not allowed to enter the cell because the ion channels that would allow sodium to enter remain closed. In addition to the fact that the neuron is basically only permitting certain ions to come inside, there are a few other things that are happening. First of all, we have electrostatic attraction. So basically, this is the idea that opposite charges attract and like charges repel each other. And basically, because of the way that opposites attract, all of the ions except for sodium are kind of held in check, along with concentration gradients. Areas that have high concentrations of ions are going to want to spread out within a medium. And one of the things that we find is that except for sodium, the electrostatic attraction and the concentration gradients basically counteract each other for chlorine, for potassium, for chlorine and potassium. The only one that this doesn't work for is sodium. Sodium has both its concentration gradient and electrostatic attraction wanting to force it inside, but the neuron is basically closed to sodium at rest. So here's a little bit more about the ion concentrations within the neuron. So here we have the inside of the neuron in blue, and here we have the outside of the neuron. As you can see, we have a few different, we have a few different players here. So we have sodium, we have potassium, we have chlorine, we have calcium, which isn't as important here, and we have these negatively charged, larger organic ions that are largely concentrated, as you can kind of see here, inside of the neuron, but they're usually too big to cross the membrane. So as you can see, there's more sodium outside of the cell, there's more potassium inside of the cell, and there's more chlorine outside of the cell. Now notice in the case of potassium, potassium is positively charged, so it wants to flow inside of the cell. However, that is completely counteracted by its concentration gradient. There's already more potassium inside of the cell, so all of the potassium basically is held in place. Now here's chlorine. We have more chlorine outside of the cell than inside of the cell. Concentration means that it wants to flow inward, but electrostatic attraction basically holds chlorine in place. Chlorine is negatively charged and the inside of the cell is negatively charged. So electrostatic attraction with chlorine is basically counteracting concentration. And again, as I mentioned, you've got more sodium outside, wants to flow inside. Sodium is positively charged. The inside of the cell is negatively charged, but notice that the ion channels for sodium are closed at rest. Now we do have things like the sodium-potassium pump, which can allow sodium inside, but it's always going to make sure that the concentration leaves more sodium outside and more potassium inside. So the sodium-potassium pump can also contribute to this resting membrane potential. Now, as I kind of mentioned, there are other things that contribute to this balance. The voltage-gated sodium channels are closed at rest. If you get a slight depolarization, and depolarization basically means that we're making the cell less negative. In this case, we're moving from -70 to -50. That's when that channel will open. But at rest, it's closed. We also have the sodium-potassium pump. It moves 3 sodium outside for every 2 potassium inside. As a result, the inside of the cell will stay negative and will still get that contribution to the resting membrane potential. So one of the things that you're going to kind of see is that action potentials will ultimately only fire if the threshold is reached. So you can kind of see an example of this here. We use a small microelectrode to inject some type of current. And depending on the nature of the current, we may either get a depolarization, such as you see here where the cell becomes less negative, or we get what is called a hyperpolarization, where the cell becomes more negative. So here you can kind of see the stimulus that we're using. We're using a particular current. It's either going to be depolarizing or hyperpolarizing. Now, here's what's kind of critical. A depolarization usually will happen either because of an influx of sodium or an influx of calcium. Both carry a positive charge. And what you can kind of see is that you do get a small depolarization, but because it isn't reaching that threshold for the voltage-gated sodium channels to open, nothing really happens. Ultimately, the potential will return very quickly to rest. Now, likewise with a hyperpolarization, this will happen either because of potassium leaving the cell or chlorine entering the cell. So you can see that the membrane potential temporarily becomes more negative before once again returning to rest. Now, in this case, we get a depolarizing current that is enough to reach threshold. And at this point, an action potential occurs. The voltage actually shifts from negative all the way up to positive before very quickly hyperpolarizing and then returning to rest. Now, one thing that I want you to kind of notice is that the stimulus here is varied in size. It gets larger and larger and larger. Notice that despite the fact that the stimulus is getting larger, the action potential does not change. This is what is called the all or none property of action potentials. Action potentials will fire exactly the same way every time, regardless of the strength of the stimulus. If it's enough to reach threshold, you're going to get the exact same action potential every time. So let's talk a little bit more about this. So in addition to neurons depolarizing and basically becoming less negative due to sodium flowing inside of the cell or calcium flowing inside of the cell and becoming less negative, neurons can also hyperpolarize. So if chlorine enters the cell, what we would call an influx, the inside of the cell becomes more negative. This is inhibitory in nature. It makes it harder for the cell to fire. Now compare that to depolarization, which can make the cell more likely to fire, particularly if that threshold is reached. Now, this will also happen if potassium leaves the cell and takes all of its positive charge with it, which is also referred to as a potassium efflux. Now, these have special names. So part of the reason for this is because neurotransmitters, basically when you take certain types of drugs, these neurotransmitters are going to act on postsynaptic membranes, basically receiving neurons. So we have a neuron that's sending a message. The postsynaptic membrane is the cell that is receiving the message. So we often refer to these as postsynaptic potentials. We have what are called excitatory postsynaptic potentials, or EPSPs. Again, these are depolarizing local potentials. So remember that they will be related to stimulus strength, but what's really critical here is that if they don't reach threshold, they're very quickly going to return to rest. And notice too that, as I said, local potentials are related to the strength of a stimulus in a way that the action potential is not. Here we have a hyperpolarizing current, and here's an even stronger one. Notice that with the first current, it gets a pretty sizable hyperpolarization. When we have an even stronger hyperpolarizing current, the hyperpolarization becomes even larger. So these are local potentials. Local potentials are related to stimulus strength. If it's excitatory, we're going to get a depolarizing current that can potentially get the cell to reach threshold and thus fire an action potential. We also have those inhibitory postsynaptic potentials, which create hyperpolarizations. So EPSPs create depolarizations, IPSPs create hyperpolarizations. So here are the stages of the action potential, and here you can kind of see why the threshold is what it is. So basically at rest, you can see that the sodium channels are closed. If we have a depolarizing current, what we are basically going to see is that some of the sodium channels are going to begin to open. But once that depolarization hits threshold, all bets are basically off. Our sodium channels are beginning to open, and sodium is going to flow inside of the cell and carry a lot of positive charge with it. Basically, to the point where the voltage of the cell has shifted from being negative to positive. But once the cell kind of reaches this positive state, that's when the sodium channels are very slowly going to become inactivated and begin to close. And because of that, no more sodium can really enter the cell. And that's why we get this peak at positive 40 millivolts. These channels are inactivated and sodium can no longer enter. At the same time, the potassium channels are beginning to open, and potassium is leaving the cell, taking all of that charge with it. So here we have the rising phase. This is due to sodium entering the cell. Here's the falling phase, and this is because of potassium leaving the cell and carrying all that positive charge with it. And in fact, because the potassium channels are very slow to close, we actually get a period where so much potassium has actually left the cell that we have a temporary hyperpolarization until all of the channels close, reset, and then very quickly we return to that resting membrane potential. Now, the action potential is basically all or none, whereas there is a relationship between the amount of stimulation that you get and the size of a local potential. There is no relationship between the size of the action potential and the amount of stimulation needed to produce it. If it's enough to get to threshold, it will always fire the exact same way. Now, the action potential is going to propagate down the axon. So something that I will mention going back for a second, around stage 4 and stage 5, the channels are closed. And this basically means that the neuron, this portion of the axon and these ion channels are basically resetting. At this point, this portion of the axon is said to be in a refractory period. No stimulation will produce an action potential at this point. And because of the refractory period, the action potential cannot move in both directions down the axon. Because the previous section of axon is still in its refractory period, the action potential does not back propagate. The action potential always moves in one direction, starting at the axon hillock near the soma and basically terminating at the terminal buttons where neurotransmitters are released. Now, some drugs ultimately can change this conduction. So for example, if you go to the dentist, you might receive a local anesthetic such as Novocaine, lidocaine, or Penocaine. And we know that local anesthetics basically create a numbing sensation because they block the opening of sodium channels. And because of this, action potentials cannot happen. And that's why when a dentist is basically drilling into your tooth and you've been injected with these local anesthetics, you won't feel anything because no action potentials can happen at this point. Now, on the other hand, we have things like saxitoxin, which is present in red tide. If you live in places like Florida during certain parts of the year, red tide can be pretty common. And generally, they will tell you not to eat any fish or seafood that may have come from areas that have red tide. And the reason for this is because saxitoxin basically blocks all of the sodium channels in the nervous system, because you've ingested it. And because of that, no action potentials will happen, and people who eat red tide infected food can potentially die by suffocation. So a couple of other things that I want to mention that would be really helpful. There are 2 different refractory periods that I didn't really mention. So we do have an absolute refractory period. In this stage, there can't be another action potential because the voltage-gated sodium channels are basically closed. Now, after the absolute refractory period, we do have something called a relative refractory period. This is that brief hyperpolarization that we see at the end of an action potential. At this point, you can have another action potential, but you're going to need a more significant depolarization. You're going to need a stronger stimulus to be able to get that. So now let's move on to talk a little bit about the basics of neurotransmitters. So here you can kind of see the little gap between neurons, the synapse that we talked about previously. So right here, we have the synaptic cleft. So here is the postsynaptic neuron, the neuron that is occurring after the synapse. And here we have our presynaptic neuron. This is the neuron that's sending the message. And in fact, you can see very tiny vesicles which basically store packages of neurotransmitters that will ultimately fuse with the cell membrane at this region right here called the postsynaptic density, and those neurotransmitters will be released into the synapse. Now, additionally, the postsynaptic neuron will have a lot of different neurotransmitter receptors so that these neurotransmitters can make contact with receptors and ultimately send that message on. So I want to talk a little bit about the general life cycle of neurotransmitters before talking about what happens when neurotransmitters are released into the synapse. So I mentioned already, neurotransmitters are basically going to be packaged into vesicles. And this is going to be really important because neurotransmitters, if they are not stored in these vesicles, they can be destroyed due to free radicals and enzymes that are circulating inside of the neurons. So vesicles provide protection from neurons. Basically, what's going to happen is that you have an endosome that's basically going to bud off and basically form a vesicle that can be filled and packaged with neurotransmitters. At some point, as an action potential moves all the way down to this axon terminal or the terminal buttons, calcium is going to flow inside of the cell. And calcium is going to be really critical here because it allows this vesicle to basically dock with the cell membrane, and it's basically going to fuse with the cell membrane through a process of exocytosis. So basically the vesicle is going to dock, it's going to prime, it's going to begin to fuse with the cell membrane, forming an omega figure, and at that point, neurotransmitter molecules are going to be released into the synapse. Then endocytosis will occur. We'll get a complete fusion with the cell membrane, but part of it is going to bud off and again join with that endosome. The neurotransmitters will bind to different types of receptors, and then it is going to be broken down. Now, one thing that I will mention, there are 2 different ways to deactivate neurotransmitters. We can do a process that is referred to as reuptake, and we can also do what is called breakdown by enzymes. Your textbook refers to this as catabolism. So here we have some neurotransmitters. They bind with a receptor site, and in this particular case, they're broken down by enzymes, or they can be picked back up by a transporter and moved back into the presynaptic cell for additional recycling. This can also happen within glial cells before being transported to the presynaptic cell. So neurotransmission occurs when neurotransmitters bind to postsynaptic receptors. So postsynaptic receptors basically are located in cell membranes, usually on the dendrites of a neuron. And basically the transmitter is going to bind to the receptor to activate it. And the resulting effect may be excitatory or inhibitory. It depends on the neurotransmitter that is binding, and it also depends on the ions that it's acting upon. Now, one thing that I will mention, in addition to postsynaptic receptors, your textbook briefly talks about what are known as autoreceptors. So oftentimes, we're going to talk about postsynaptic receptors, but there are what are known as presynaptic receptors. Autoreceptors are basically receptors that occur on the membranes of a presynaptic cell. Autoreceptors are often designed to decrease neurotransmitter release. So they're kind of there as a negative feedback loop. So if you're making too much, if you're a presynaptic cell and you're making too much of a neurotransmitter, the autoreceptors will pick that neurotransmitter up and basically reduce the amount of neurotransmitter that is being released. But we are largely, for the purposes of this class, going to talk about those postsynaptic receptors, but do be aware that we do have presynaptic receptors like autoreceptors, and drugs can potentially affect these as well. Now, there are two main types of receptors that we're going to talk about. Ionotropic receptors, which are shown here, and metabotropic receptors, which are shown here. Now, ionotropic receptors, basically this is an ion channel that will open when a neurotransmitter binds to the receptor, like so. So here we have our ions in red. When the neurotransmitter binds with this receptor, the ion channel opens, allowing ions to flow inside of the cell. Now, neurotransmitters are technically ligands, and as a result, the ionotropic receptors are what are called ligand-gated receptors. Now, metabotropic receptors are going to be a little different. A neurotransmitter is going to bind, and then at that point, things like G proteins will convey messages to the rest of the cell, often through use of enzymes, protein kinases, and second messenger systems. Now, I want to talk a little bit more about the difference between ionotropic receptors and metabotropic receptors. And your book has a really helpful chart in table 3.1 to help you out with this. So ionotropic receptors are going to be attached to an ion channel that's embedded in the neuron. Now, compare that to a metabotropic receptor, which is ultimately kept separate from ion channels and from other proteins. We've already kind of talked about the major effect that both of these receptors do. Now, ionotropic receptors are pretty fast acting. They will typically end their functioning when neurotransmitters leave the binding site. So basically, when neurotransmitters no longer bind to that ionotropic receptor, the channel closes. Now, in the case of a metabotropic receptor, we do have effector enzymes that are basically going to engage in a cascade of events that are going to persist long after the neurotransmitter is no longer binding to that metabotropic receptor. Generally, the impact of neurons with an ionotropic receptor, we're going to get an influx of ions. It's going to potentially change the local potential. We'll either get an EPSP or an IPSP. In the case of a metabotropic receptor, you can get a local potential as well, but there are going to be other types of cellular effects. That includes things like regulating enzymes, expression of genes, and the synthesis of proteins. So I want to talk a little bit more about metabotropic receptors. So basically, here's our metabotropic receptor. PowerPoint kind of cut off the image here, but here's our neurotransmitter. It's basically binding with this receptor. So basically, once this is bound, once the neurotransmitter has bound to this receptor, the G protein is basically going to send a message to this effector enzyme. It is the job of the effector enzyme to basically release different types of second messengers. So effector enzymes could include things like phosphodiesterase, which is a very critical enzyme involved in the depolarization, or rather the local potentials of rods and cones that enable us to see. So this effector enzyme is going to release a second messenger that can include things like cyclic GMP. And that second messenger is going to activate a protein kinase. This is going to cause phosphorylation. Phosphorylation is basically when we add a phosphate group, but it can cause a variety of other cellular effects. So you can kind of see why ionotropic receptors tend to be much faster than metabotropic receptors. You have this whole cascade of effects. Basically stimulating a G protein, going to activate an effector enzyme, going to release a second messenger, going to activate a protein kinase, going to engage in a variety of different cellular effects. So you can see why metabotropic receptors tend to be a little bit more slowly acting. Now, there are different types of neurotransmitters, and we're going to talk about those now. Your textbook does spend a little bit more time talking about endorphins and gases like nitric oxide. So make sure that you take a look at those. I'm going to cover some of the bigger ones here. So there are several different neurotransmitters that are involved with psychoactive drugs, and we're going to talk about quite a few of these today. I would also encourage you to read about these in your textbook, and we're going to go over each of these in detail. So we're going to talk about glutamate, GABA. We're then going to talk about the monoamines. We're going to talk about the 3 catecholamines, dopamine, norepinephrine, and epinephrine. And then we're going to talk about the indolamine, serotonin. We're also going to talk about acetylcholine, and I would encourage you to read on endorphins and nitric oxide on your own time. So we're going to start by talking about glutamate. So glutamate is an excitatory amino acid neurotransmitter. So basically, anytime glutamate binds, you're going to get excitation. You're going to get EPSPs and action potentials, and a greater likelihood that neurons are going to fire. Now, glutamate is actually present in many different proteins, and it's responsible for many different metabolic functions, and all neurons in your brain and all glial cells contain it. The creation of glutamate, it is synthesized from glutamine. So here you can kind of see the chemical structure of glutamine. And then with the help of an enzyme, remember that sometimes to synthesize neurotransmitters, we're going to need a precursor and we're going to need an enzyme. So glutamine is the precursor for glutamate. The critical enzyme here is glutaminase, and here is the chemical structure for glutamate. So when we talk about glutamate, we have ionotropic receptors and we have metabotropic receptors. So the ionotropic receptors are going to allow for very fast glutamate action. Now, your book does mention a few of the metabotropic receptors, but we are largely going to talk about these 3 receptors. We're going to talk about the AMPA receptors, the kainate receptors, and the NMDA receptors. So what do the ionotropic receptors do? They basically depolarize postsynaptic cells and thus generate an excitatory response. So here you can see the AMPA receptors and the kainate receptors. Both of these are sodium or ligand-gated sodium channels. What's kind of interesting about NMDA receptors is that while they are ionotropic, they act in some ways like a metabotropic receptor. So here with NMDA, glutamate is going to bind. That allows both sodium and calcium to flow inside of the cell, and calcium basically is going to act as a second messenger and create a lot of different cellular effects. So it's kind of an ionotropic receptor, but it's also kind of a metabotropic receptor. So as I kind of mentioned, NMDA receptors have some really interesting properties. They don't act like most ionotropic receptors. So I mentioned that they have to be opened by glutamate, but they also need to be opened by glycine. Glycine is another inhibitory neurotransmitter that we're not really going to talk about here. If glycine does not work, D-serin will do the job. And in addition to this, there are binding sites that may block ions from entering. So those of you who have had me in biopsych have already heard that NMDA receptors have a magnesium block. The cell needs to slightly be depolarized so that magnesium can basically move and those ions can enter. But you may not know that NMDA receptors also have a site for binding to PCP, which we will talk about later on in the semester. So how do we deactivate glutamate? It is going to largely be removed by glutamate transporters. And these are called excitatory amino acid transporters, or EAAT. I like to call it EAT. But basically, they're going to be, after binding with a receptor site, these transporters are going to move it back into the presynaptic cell, and glutamate can then be recycled. Now we're going to talk about GABA. And you might be kind of interested as to why I am talking about GABA after glutamate. Well, here's why. GABA is an inhibitory amino acid neurotransmitter. So glutamate was the excitatory version. GABA is the inhibitory version. GABA is short for gamma, this is the Greek gamma, aminobutyric acid. And one of the things that is really important here, inhibition is just as necessary for us as excitation is. Excitation in too large amounts can be dangerous for us. Our brains need inhibition. We have found through animal models and through work with humans that if we block GABA, people may experience convulsions and may even die. The cells become overexcited and they become so excited that they end up dying as a result. So we want to protect our brains, and our brains thus need inhibition. So GABA is just as important as glutamate is. Now, GABA is basically synthesized from glutamate. So we use glutamate to make GABA. So here's glutamate. With the enzyme glutamic acid decarboxylase, or GAD, we get GABA aminobutyric acid, or GABA, and this takes one single step. There are two major classes of GABA receptors. We're not going to spend too much time on these. We have the GABA A receptor, which is ionotropic, and then we have the GABA B receptor, which is metabotropic. Now, what I think is kind of interesting, you're looking at a GABA A receptor here. There are not only binding sites for GABA, but there are different sites on this receptor where different types of drugs will bind, including things like picrotoxin, benzodiazepines, neurosteroids, and barbiturates. Now, what happens after reuptake of GABA? So much like glutamate, is subject to reuptake. GABA is as well. GABA is basically broken down once it's brought back into the cell. We have an enzyme called GABA aminotransferase, or GABA-T, and this creates glue, which can then be used in the form of glutamate, or it can be used to produce more GABA. Glutamate is then going to be converted into glutamine by astrocytes, and at that point, you can reconvert it in GABAergic neurons. So now we're going to talk about the monoamines. We have dopamine, norepinephrine, epinephrine, and serotonin. And we're going to start with dopamine first, and in a couple of slides, you'll see why we started with this one. Dopamine is ultimately the first neurotransmitter that is synthesized. So here you can kind of see we're going to start with our precursor. So we get tyrosine. Tyrosine is basically the precursor to dopamine, but what's important is tyrosine comes from phenylalanine, which is present in your diet. With phenylalanine, you then get tyrosine. And then with the enzyme tyrosine hydroxylase, we then get dopamine, or we get dopa. This is referred to as levodopa. If you are taking medication for Parkinson's tremors, you might receive levodopa because dopamine is too big to cross the blood-brain barrier through a pill form. So oftentimes people with Parkinson's will be given dopa, or sometimes referred to as levodopa, as the precursor. So we start with tyrosine, we then get levodopa, and then we have aromatic amino acid decarboxylase, which then creates dopamine. Dopamine can then be used to create norepinephrine and then epinephrine. Dopamine is basically converted into norepinephrine with dopamine beta hydroxylase, and then norepinephrine can then be made into epinephrine. So converting tyrosine to dopa is what takes the longest step. Tyrosine hydroxylase is basically what's called the rate-limiting enzyme. So basically, how much dopamine and norepinephrine you are able to produce ultimately depends on how much tyrosine hydroxylase you have in your system. So this is what's called a rate-limiting enzyme. It's basically going to determine the rate of dopamine and norepinephrine production. So we're going to talk a little bit about the dopamine receptors. So dopamine uses 5 different receptor subtypes. We go D1 through D5. All of them are going to be metabotropic. We have what is called a D1 family, and we have what's called a D2 family. D1 and D5 are going to be a part of the same family. D2, D3, and D4 are going to also be a part of the same family. And as you can kind of see here in this figure, this kind of implies that this G protein has an excitatory effect. This G protein has an inhibitory effect. So what we know is that the D1 family is generally going to be associated with excitatory effects. The D2 family is generally going to be associated with inhibitory effects. So we will often deactivate dopamine by catabolism, by that enzymatic breakdown. And there are 2 different ways that we can do this. We can either have dopamine broken down by catechol O methyltransferase, or COMT, or monoamine oxidase, or MAO. For those who may not know, MAO inhibitors may occasionally be used to treat clinical depression. We basically prevent this enzyme from breaking down dopamine, norepinephrine, and serotonin. MAO inhibitors include drugs like phenelzine, which is sold under the trade name Nordal, and phenylcypromine, which is sold under the trade name Parnate. Now, as we get into more more complex functions of these neurotransmitters. We didn't really need to talk about the glutamate pathways in the brain because glutamate is in every single neuron and GABA is as well. But when we start talking about acetylcholine and dopamine and serotonin and norepinephrine, we have distinct pathways that are going to be kind of critical. So the dopaminergic pathways, we have 4, but I'm going to talk about 3 major ones that are critical for humans. The dopaminergic pathways originate in several different cell groups, either in the brainstem, typically in the pons, as well as in areas like the ventral tegmental area of the midbrain. And we have 3 major ascending dopaminergic pathways. We have the nigrostriatal tract, the mesolimbic tract, and the mesocortical tract. So the nigrostriatal pathway arises in the substantia nigra, and it basically projects to the basal ganglia. The mesolimbic starts in the ventral tegmental area, and it terminates in the limbic system, particularly in an area called the nucleus accumbens. The mesocortical tract also starts in the ventral tegmental area, and it terminates in the prefrontal cortex. We have another pathway that is called the tuberoinfundibular pathway, that starts in the hypothalamus and basically terminates in the pituitary gland. But we're not really going to talk as much about the tuberoinfundibular pathway. So again, here are the tracts. Here you are looking at these pathways in the rat. I will show you the pathways in humans shortly. The nigrostriatal tract is really critical for voluntary movement. You can see here we start in the substantia nigra and we terminate in the areas of the basal ganglia, including the caudate, the putamen, and the globus pallidus. In Parkinson's disease, one of the things that we find is that there is a loss of dopaminergic neurons in the substantia nigra, and as a result, we have difficulties with movement. Here's the mesolimbic tract. It starts in the ventral tegmental area and it terminates in the limbic system, particularly in the nucleus accumbens. This tract is going to be really critical for processing things like reward, as well as basically kind of functioning, for better or for worse, as an addiction circuit. And then finally, we have the mesocortical tract, again, starting in the ventral tegmental area and basically terminating in areas of the cerebral cortex. This pathway is going to be really important for things like planning, memory, and it also has, like the mesolimbic pathway, some implications in addiction and reward. And here's what they look like in humans. So here we have our nigrostriatal tract. Here we have our mesolimbic tract, here is our mesocortical tract, and then here is our tuberoinfundibular tract. Now we're going to move on to talk about epinephrine and norepinephrine. Now, one of the things that I will mention is that epinephrine is more often used in the peripheral nervous system than in the central nervous system. So for much of this class, we're largely going to be talking about norepinephrine and the noradrenergic system. So let's talk a little bit about some of the general characteristics of epinephrine and norepinephrine. They also are known by the names adrenaline and noradrenaline. And these are going to function in both the central and the peripheral nervous system. Norepinephrine is the primary neurotransmitter of the sympathetic nervous system, as we talked about last time. The cell bodies that basically produce noradrenergic neurons are found in the brainstem, in particular the locus coeruleus, which is a collection in the pons. Now, compared to the dopaminergic pathways, there is a widespread distribution of noradrenergic pathways, particularly when we're talking about the forebrain and the cerebral cortex. So here you can kind of see the noradrenergic system in the rat. Here's our locus coeruleus. We have terminations in the cerebellum, the pons, the medulla, the midbrain, the inferior and superior colliculus, the thalamus, the hypothalamus, the limbic system, and the cortex. So you can see that we have a significantly more widespread distribution here than we did with our dopaminergic pathways. And you can see this in humans as well. So here is kind of our locus coeruleus right here, and you can see this widespread distribution throughout the brain and the cortex. Now, norepinephrine has many different behavioral effects due to its widespread distribution. Now, because it is the primary neurotransmitter of the sympathetic nervous system, we know that it's going to affect a variety of different functions that are critical when we are in fight or flight. So this will include things like arousal, our sense of alertness, attention, sleep-wake behaviors, cognition, and also storing emotional memories. Now, there are many different noradrenergic receptors. There are two major classes. They're alpha and they're beta, and all of them are metabotropic. Now, if you are struggling with severe anxiety or panic attacks or high blood pressure, you may be given what is referred to as a beta blocker. The reason that we call these beta blockers is because these drugs block beta receptors for norepinephrine, as a result, reducing those kind of fight or flight effects. We know that blood pressure can rise under a fight or flight response. We know that anxiety happens as a fight or flight response. So beta blockers prevent those things from happening. Now, they are going to be very similar to dopamine receptors in this regard, and their catabolic enzymes and their reuptake mechanisms are also pretty similar to dopamine. One thing that I didn't mention is that reuptake can happen for dopamine as well, but it's not going to be talked about as much as the catabolism, that breakdown by enzymes. Now, let's talk a little bit about serotonin. So serotonin, unlike, I want to go back just to kind of show you something kind of critical. So if you look at dopamine and norepinephrine, you'll notice that both dopamine and norepinephrine, and a picture is not shown here, but epinephrine has this as well. All three of these have this nice little catechol ring. And it's for this reason that dopamine, norepinephrine, and epinephrine are referred to as catecholamines. Now, serotonin does not have a catechol ring. So because of that, serotonin is sometimes referred to as an indolamine. Now, in this case, you can see its precursor is tryptophan. So tryptophan, again, is going to be largely from our diet, as is phenylalanine and tyrosine for our catecholamines. So tryptophan is going to be contained in protein-rich foods. As a fun fact, you do need to also have carbohydrates in your diet to basically create this synthesis from tryptophan all the way to serotonin. So tryptophan is converted into 5-hydroxytryptophan, or 5-HTP, through tryptophan hydroxylase. Tryptophan hydroxylase is the enzyme that does this conversion, and it is going to be the rate-limiting enzyme for serotonin. Once you have 5-hydroxytryptophan, the enzyme that's going to be critical here is 5-HTP decarboxylase, which basically creates 5-hydroxytryptamine, which is also 5-HT or serotonin. Now, the serotonin system, or the serotonergic system, is also going to be very widespread, as you can see here in the rat. Now, the serotonergic cells in the central nervous system are found in a large network in the brainstem, particularly in the dorsal and the medial raphe nuclei in the pons. And as you can see, we have a lot of different inputs here. We have input to the cortex, the striatum, the nucleus accumbens, the limbic system, the cerebellum, the thalamus, and the hypothalamus. And again, this is similarly widespread in humans. Here's the raphe nuclei, and you can see that widespread distribution to areas like the cerebellum, the hypothalamus, the thalamus, the basal ganglia, the frontal cortex, and the limbic system. So finishing up, we're going to talk about acetylcholine. So acetylcholine is kind of different from the other the monoamines that we talked about. It has a pretty simple chemical structure, and it's made up of two separate components. It's made up of choline and acetyl coenzyme A or acetyl-CoA. As with serotonin and norepinephrine, acetylcholine is diffusely distributed throughout the brain. And this is going to be a critical neurotransmitter for muscular movements. It is because of acetylcholine that the muscles in your body are able to contract, and it's also the primary neurotransmitter of the parasympathetic nervous system. So much as we saw with GABA, the synthesis of acetylcholine is very rapid. Only one single step is going to be involved here. So we have choline, we have acetyl-CoA, and then we have our critical enzyme, choline acetyltransferase or CHAT, and then we get acetylcholine. Now, there are two major classes of cholinergic receptors. I did forget to mention that serotonin has about 7 different types of metabotropic receptors. We're not really going to talk about them very much, but with acetylcholine, we have 2 major types. We have what are called nicotinic receptors. These are ionotropic in nature. And then we have muscarinic receptors, and these are metabotropic. So nicotinic receptors ultimately respond to nicotine, which is an agonist of the nicotinic receptors. It facilitates the effects of acetylcholine. And then we have muscarine. Muscarine is present in the fly agaric mushroom, and that is also going to be an agonist of the muscarinic receptors. So acetylcholine is broken down by acetylcholine esterase, or ACE. It's going to break acetylcholine down into choline and acetic acid. So this catabolism is basically going to terminate neurotransmission. And basically, acetylcholine esterase is basically going to metabolize acetylcholine in the synaptic cleft. And that's largely what's going to be done. Now, what sort of things is acetylcholine good for? I've already mentioned a few of these. It is the primary neurotransmitter of the parasympathetic nervous system. It's released at the neuromuscular junction, so the whole reason that you can contract your muscles is because of acetylcholine. And it also plays a role in different facets of the central nervous system. So here's the cholinergic system in a rat brain. So you can see that we have some areas like the striatum. This is going to be critical for movement, and the balance between dopamine and acetylcholine is going to be really critical here. We also have areas like the basal forebrain, which is going to be critical for attention and learning. Now, here's what this looks like in humans. So we have these 3 major pathways. The first one kind of begins in the pons and terminates in the midbrain. This is going to be critical for sleep-wake behavior. We have another one that is critical for learning and memory. And then a third one around the septum, which is critical for autobiographical memory. Now, your textbook is going to talk about other types of neurotransmitters and other types of chemical transmission. So make sure that you're kind of reading about neuropeptides, nitric oxide, and then other types of chemical transmission, particularly hormones. But folks, this is all I had. This one was a little bit longer, but I think you will be in good shape to talk about some of the different drugs next week and how these affect the different types of neurotransmitters that you've learned about today. So that is it, and thank you so much. I will see you next week with more lectures.